I learned recently that when the Nobel Prize in Chemistry was announced earlier this month, that some chemists in Kentucky were really excited about this year's winners. So I talked with them and wanted to share that with you, but first some background. This year's Nobel Prize in Chemistry was awarded to three scientists: Morten Meldal of the University of Copenhagen in Denmark, Barry Sharpless of Scripps Research in California, and Carolyn Bertozzi of Stanford University. This year's prize honors the remarkable progress that's been made in what's known as click chemistry and bioorthogonal click chemistry. Click chemistry is essentially a more efficient way to join molecules together. Meldal and Sharpless are credited with getting the ball rolling on click chemistry, and Bertozzi is credited with taking it to a new level and developing click reactions that work inside living organisms. Here's what scientists who are members of the Kentucky Academy of Science had to say about this year's Nobel Prize winners in chemistry.
This particular award is such a really slick piece of organic chemistry. One of the challenges to organic chemistry is creating bonds, and that can be a really difficult thing to do. I mean, a lot of times you need very stringent conditions. A lot of the techniques that chemists use may use very strong bases. They may require anhydrous conditions, or other really aggressive and carefully controlled air sensitive conditions and things like that in order to build chemical bonds. And I think part of the genius of the click chemistry is this idea that you can take these things and put them together under really mild conditions, and really, I would say, even the idea of the bioorthogonal reactions that Bertozzi envisioned or articulated, this idea that you could actually do an organic reaction not just in water, which is hard enough, but that you could actually do it in a cell, and that you could have this reaction not bother all the other cellular processes that are going on. That's really an ingenious way to approach things, and the capability of it, has been made evident over the last few years in terms of all the different applications in, particularly in molecular biology.
The click chemistry is should be able to give us more synthesis tools that can lead to better drug molecules. I mean, hopefully it'd be cancer. It could be Alzheimer's medicines. It could be a lot of different therapeutic areas.
One of the things that it has where it's seen a real use is molecular, or I would say cellular visualization, understanding how cells are behaving and tracking changes, particularly, for example, on the surface of cells, by attaching probes, things that we can see specifically to certain types of cells. We can monitor how these different cells change, how their surfaces change, even through, for example, at different stages in development. One of the really interesting applications of it is in cancer treatment. And essentially the trick is that, a lot of chemotherapeutics, a lot of cancer drugs are so devastating to the body. If there's a way that you can target a particular area. So one of the strategies that has been employed is to actually inject sort of a receptor right in the vicinity of a tumor that is being treated, and then introduce the cancer drug. Well, that drug can float around benignly or circulate through the system, but once it hits that area where the tumor is, then it will lock on, activate, and actually release that drug in a selective place. So studies have shown that cancer that using this approach can drastically reduce the side effects from cancer treatment. So that's one of the exciting applications.
The thing that's interesting about Bertozzi is she was actually doing the pre-medical path, the medical path. And that's the way most students are. They have a path, and it's kind of like following the yellow brick road. And if you stay on the path and you don't get off the path, you're going to get this job. You can live in this place and make this amount of pay. And that's the way most people are. Well, Bertozzi, basically, she was taking organic chemistry at Harvard and said, I love organic chemistry and this is interesting. And so she got off the yellow brick path, and she just kept taking opportunities and just kind of following her heart and what she thought was interesting. And she really has done some unique chemistry with sugars. And sugars is really the future of organic chemistry and a lot of the biological mechanisms, I believe. There's so much chemistry that's unknown. And so sugars, we have lots of variety of sugars. We have glucose, xylose, fructose, and then we have these modified sugars. And so in our brain, if we want to remember something, it's stored in a sugar polymer. And it's just like our antibodies that recognize our red blood cells. So we have type A, type B, and type O type of blood cells. And it's basically the way the polymer sugars are connected to each other. And so we are limited in our understanding of organic chemistry and how to resynthesize these sugars. What Bertozzi focuses on is a thing called bioorthogonal. And bioorthogonal is a mathematic term that means perpendicular. And so they're not parallel, they're perpendicular. So if they're bioorthogonal, that means those functional groups don't interact in biology. And so she did something very simple. She took two functional groups that you don't find in our biological systems, an azide and an alkyne. And she said, and these react really quickly and specific to make a triazole, a 5-membered ring. And so anytime they come into contact, they're gonna click, like your seatbelt clicks. And so you can take one molecule, and you take another molecule, and then when they come together, because they're not gonna run into any other biological molecules, they click. And now you have labeling and you can see things. Sometimes it's just being on there, seeing the right opportunity. And that's what she talks about. You know, you just, when you think that's good, and take that opportunity. And she talks about taking paths that are unknown. You just follow your heart and what's interesting.
Right.
And I think that's what we need for science.
You know, the idea of being able to run an organic synthesis reaction at such high yields, so reliably, not just in an aqueous solution, but even in a cell, is just extraordinary.